Welcome to Zach D TV, the place for interesting news from around the net. In today's episode, we're going to look at two amazing medical advances and an interesting study. And if you want more interesting news 7 days a week, go ahead and click that subscribe button right over here so you know when I upload something new. Alright, let's get into this. On Monday, researchers from the Houston Methodist Research Institute released a paper in the Journal of the American College of Cardiology that outlines their attempts to slow aging. According to Dr. John Cook, the lead researcher in this team, we can make aged cells younger. That is a pretty bold statement. Let's get into this. What Cook's team did was take cells from 17 kids with progeria. That's a disease that makes the human cells age rapidly. It makes children look older than they actually are. A lot of times they die of heart attack or stroke before they hit 20 years old. And effectively, by that time, they look like old people. It's rapid aging, and it's a very unfortunate disease. And what they found was 12 out of the 17 patients had shortened telomeres. In fact, they say they were equivalent to the telomeres you would find in about a 69-year-old, a healthy 69-year-old. A telomere is an end cap on your chromosomes that every time the cell divides, they get a little shorter and a little shorter each time until eventually they quit reproducing and the cell dies. And you can find reports all the way back to 2010 that believe these telomeres are directly linked to aging. So what Cook's team did was directly inject RNA into the cells to spur the release of telomerase. It's a protein that leads to the growth of these telomeres. According to the paper, where the cells would multiply very poorly and then die before the injection, they found after the injection, these cells proliferated normally and released less inflammatory proteins, which would cause inflammation of joints and stuff like that. Kind of like arthritis you see in older people. So basically, it put these cells in a healthier state, in a younger state, if you would. Now, it's all well and good. This is preliminary testing, and it all happened in petri dishes. So it's not the same as having it happen inside of a human, but these are great first steps into a cure for aging-related problems. And like I said, these studies have been done before, but this is one of the first ones that showed such great success. And they hope to build off of this. And while this isn't really a fountain of youth, it may be able to extend human life longer than what we currently have, which I'm okay with. What do you guys think? Should we be messing with our timelines? Should we be extending human life? Or is it something that nature decides and we should just go with it? Let me know in the comments below. Up next, I want to talk about a story that's been in the news for the last couple days. It's making its rounds, but I wanted to wait until I saw the actual published paper before I reported anything on this. And that is that researchers at Oregon Health and Science University were able to correct a genetic defect in a human embryo. First time this ever happened here in the United States. And that's because our laws against it are very strict, and there's a lot of loopholes to jump through. While studies like this have been done, mainly in China, because of the more lax rules on working with human embryos, this one shows a lot of promise compared to the other studies. And it also used a lot more embryos than any other study like it before. The team led by biologist Shokrat Matilipov started out with 52 human embryos. And using Crispr-9, they were able to fix a genetic disease known as hypertrophic cardiomyopathy. This disease affects 1 in 500 people, and they showed success in 42 out of the 58 initial embryos. How they did this was they took DNA from a man who has this disease. In fact, he even has a pacemaker at this point. They then used his DNA to fertilize these embryos. At the same time as fertilization, they used CRISPR to cut out the flawed sequence, and they left a DNA template in with the package, so that way it'd be able to build a normal gene in its place. This is the first time it fixed every cell in the embryo, unlike previous models where it only fixed some of them. The odd part about this is that in 41 out of the 42 embryos, they didn't use the DNA template. They took that gene that was missing from the mother's side instead of getting it from the father's DNA, and it built its own replacement gene, which is still viable and works well. But part of the reason I wanted to wait on this research is because of the headlines that have been out there. You see them all over the place. Oh, designer babies are coming. It's time for designer babies. Here's proof designer babies can happen. No, in fact, if anything, this proves that making designer babies is going to be harder than we thought. Because this research shows that when a DNA sequence has an empty spot in it, it grabs its info from the mother's cells instead of building off what we want it to. So the chance of our designer babies taking on the traits that we implant into them is very, very low. Like in this situation, 1 out of 42 took that template. But no matter how you look at it, this is a great advance in genetic therapy. Now, of course, they never had any intent to let these embryos grow. They weren't implanted into a womb. They were all properly disposed of. But that brings up another ethical question. I have a lot of ethical questions when it comes to medicine. Is it right for us to grow embryos to do this kind of testing on? Because I know there's some people out there that believes life starts at conception. Or is this okay because at the point where they're disposed of, they don't even have a heartbeat yet? Let me know what you think in the comments below. And this last one, you're going to wish you didn't see. Researchers at the Clemson University of South Carolina recently did a test to see what happens when birthday cake candles are blown out. The researchers made a fake cake, laid some icing over top of some tinfoil, put candles in it, and had people blow them out. They then tested the icing for bacterial presence. They tested before and after the cake was blown out. And they found that the amount of bacteria on the cakes that were blown out was increased by 1,400%. Yeah, the act of somebody blowing out a birthday cake results in 15 times more bacteria in the frosting. Now, Paul Dawson, he's the study's co-author, does state that you will probably not get sick from this. A lot of these are harmless bacteria, the same that you'd get just from whispering to another person or talking within a couple feet of someone. So it's not really that bad. Just the thought of all that bacteria running around on top of your cake might be worse for some people than others. But of course, use common sense. If the person blowing the cake out is sick, don't eat it. But with that, I'm going to wrap it up. Thanks for stopping in. Remember, I do this 7 days a week with a live stream on Saturday and a short on Sunday. So I hope to see you here again. And until next time, have fun and be safe.